In comics, it's often depicted as somebody having their DNA changed and thus gaining superpowers and becoming a level of superhero. One particular example of this is Spider-Man. In the movie Spider-Man, we have Peter Parker, resident nerd and photographer for a high school, ends up having a radioactive spider come down and bite him.
MJ, let's go.
Wait. Thanks.
In the story, now that Peter Parker has been bitten by this radioactive spider, he undergoes a change. His old DNA ends up being replaced and is now replaced with part of the spider DNA, thus granting him superpowers. These powers include the ability to have super strength, being able to see normally or in heightened senses, as well as acrobatics and webs.
Weird.
While this is science fiction in a sense, it may not be too far off in the future. But first we have to understand how something like this could be able to give somebody superpowers, and what the modes of by changing somebody's DNA ends up leading to different proteins being expressed and different traits being expressed in organisms.
This morning, any change?
Change?
The first part of how DNA is going to be able to be changed into proteins is called transcription. Let's take a look at how it works. Remember we saw in the strawberry lab that the DNA within all cells kind of looks like a gooey booger. But this gooey booger is an actual complex molecule that stores all of your information. The question is how does it go from this gooey booger into these proteins that make your eyes blue, your hair certain colors, your eyes brown, your skin color, even so far as personality and other traits? The first thing we have to be able to do is read specific parts of the DNA, as well as the proteins that they express. That's called transcription. Remember, DNA is in all cells, contains all the information for the cell, is read to make the cell function. DNA in the cell is incredibly massive, with human beings containing 6 billion base pairs per cell of DNA. There has to be a way to be able to hone down and only express certain traits that need to be expressed at certain times on the DNA. This process to be able to do that is called transcription. What is transcription? Transcription, when DNA is read and a copy of mRNA is made. mRNA stands for messenger RNA. mRNA tells the cell to perform a specific task. How does transcription work? First, a molecule called a promoter binds to the specific gene on the DNA. The gene is the task the cell needs to perform, recognized as a TATA box segment on the DNA. Here's a pictorial diagram of a segment of DNA. Notice the TATA region or TATA box region. This is the area where the promoter will bind and then have the gene expressed for the cell to perform. You can think of a gene as a specific trait, for example, the gene for eye color or the gene for hair color. RNA polymerase binds to the promoter then. RNA polymerase is a molecule that binds to the promoter and then binds to the DNA that will then create the RNA. Then it unzips a segment of the DNA. Here we see the DNA coming apart and then a C being added for the RNA polymerase. It starts to read the DNA and place a matching RNA base on the new strand. The RNA polymerase will move down the DNA then, and depending on what DNA base it sees, will add a matching RNA base, thus creating an mRNA chain that looks similar to the DNA. Matching bases. If there's a DNA A, it goes to an RNA U. If the DNA has a T, it goes to an RNA A. If the DNA has a C, it goes to an RNA G. And if the DNA has a C, it goes to an RNA G. Notice this will continue along until the ribosome or the RNA polymerase is told to stop, at which point the mRNA will disconnect, the RNA polymerase will fall off, and the mRNA goes to perform a specific function. Once RNA polymerase is done, the finished mRNA detaches and goes to the ribosome. Let's look at an animation of how transcription works so that way you can visualize it all happening in one fell process. Here we see a segment of DNA strung out along the inside of the cell. This is the unit that wants to be read, or the gene that wants to be read. First, we need to identify the TATA box, which is located with the TATA region at the beginning of the gene. The enhancer region is part of the region that the proteins will be able to bind to. In comes the promoter protein. In this case, it's TFID. It will then recognize the TATA box and bind. Now that it's bound to the TATA box, it sends out a signal and changes configuration, bringing other proteins to bind to the DNA. Once all the proteins are in place, RNA polymerase comes, recognizes the configuration, and attaches itself to the DNA. Once it is all assembled, it now can begin reading the DNA and creating an mRNA copy of it. You'll see what this ATP is later. Now the RNA polymerase is going, reading one base at a time. Notice the unzipping that occurs on the DNA where the RNA polymerase is. As it's unzipping, it is creating copies of mRNA strands to matching bases. It will then detach, and now you have a fully functional mRNA unit ready to tell the cell what to do. Now that we have transcription under our belt, the mRNA is whisked away off into the cell. But what happens next? mRNA is not what makes your eyes blue, or your hair a certain color, or gives you even superpowers in a sense. That's the next step called translation, and we'll see that tomorrow. Have a good one.